Good morning, friends. We are studying here different metabolic pathways and ATP generation during all these metabolic cycles. We discuss EMP pathway, HMP pathway, TCA cycle also, and we studied energy generation and ATP molecule generated during the metabolic cycles. We also discussed regarding the fermentation, heterolactic fermentation of lactic acid, and now we are studying differences between respiration and fermentation. You should remember that respiration is also a metabolic process in which organic compound acts as electron donor and inorganic compounds acts as electron acceptor. Whereas in fermentation, the organic compound acts as a both electron donor and electron acceptor also. Now we study different characteristics of fermentation and respiration and the differences also simultaneously. So in this particular slide, I have shown here the difference between respiration and fermentation. The first point is taken here is requirement of oxygen. So we know that respiration in case of microorganism, it will be aerobic as well as it may be anaerobic. Aerobic in the sense the organism require oxygen for their respiration. Those organisms are called as aerobic organism, or respiration is said to be aerobic type of respiration. For anaerobic organism, there is no requirement of oxygen. So in aerobic organism, respiration occurs in presence of oxygen. So they need oxygen for their respiration, whereas for anaerobic organism, there is no need at all of oxygen. But in fermentation, no requirement of oxygen strictly. Then second point is requirement of electron transport carriers and electron transport chain. So in respiration process, there is a necessity of electron transport chain, definitely. So there is a yes, electron transport chain is required for respiration. In fermentation, there is no need of this electron transport chain. Then secondly, the site of occurrence, that is where the respiration process occurs. In case of bacteria, we know that bacteria being prokaryotic cell, there is no presence of mitochondria. So as electron transport chain is located in the cell membrane, the site of respiration will be naturally cell membrane or plasma membrane, you can say. Whereas in eukaryotic cell, the respiration occurs in mitochondrial membrane. Because eukaryotic cell possess mitochondria, the prokaryotic cell do not have mitochondria. So in bacterial cell or in prokaryotic cell, the electron transport chain is located in the cell membrane itself, and therefore respiration occurs in cell membrane. Mitochondria is present in eukaryotic cell. Therefore, eukaryotic cell respiration occurs in mitochondrial membrane, whereas fermentation occurs in cell cytoplasm. Then regarding the ATP molecules, in case of respiration, if we count total number of ATP, there will be 38 molecules. We know in EMP pathway 8 ATP, in TCA cycle 24 ATP, so 24 plus 8, 32, and 6 from pyruvic acid oxidation, so 38 molecules will be there. Then in case of fermentation, if we see total number of ATP molecules, if we calculate, it will be low, so approximately 2 ATP molecules only from one glucose molecule, there is a generation of ATP. So in respiration, and fermentation. The basic difference is that respiration, it is also energy generating metabolic process in which organic compound acts as electron donor, inorganic compound acts as electron acceptor. In case of fermentation is organic compound acts as a both electron donor and electron acceptor. Regarding the oxygen requirement, respiration may be aerobic or anaerobic. Some organism may be aerobic, some organism may be anaerobic. So for aerobic respiration, oxygen is strictly needed, whereas anaerobic organisms do not require oxygen. In fermentation, there is no need at all of oxygen. Then regarding the site of occurrence where the respiration occurs, in case of respiration, in case of prokaryotic organism or in bacterial cell, it occurs in plasma membrane, as the site or electron transport chain is almost located in cytoplasmic membrane. In case of bacterial cell, there is a respiration. In case of eukaryotic cell, it occurs in mitochondrial membrane. Because in eukaryotic organism, mitochondria are there, so mitochondrial membrane is the site for respiration. The fermentation occurs in cell cytoplasm almost. One thing you remember that in case of bacterial respiration or aerobic respiration, terminal electron acceptor will be always molecular oxygen. Oxygen is there. In presence of oxygen, respiration occurs. Therefore, oxygen will be terminal electron acceptor in aerobic organism or aerobic respiration. In case of anaerobic organism, other than oxygen, there will be electron acceptor. There may be carbonate, there may be sulfate, there may be nitrate in anaerobic organism because it happens in case of in absence of oxygen. So there is no oxygen. Terminal electron acceptor will not be oxygen. Instead of oxygen, there will be either carbonate or sulfate or nitrate. This is one difference. And regarding the ATP molecules, in respiration, high number of ATP molecules will be observed there, whereas in fermentation, only 2 molecules of ATP will be generated per 1 glucose molecule. Then now we are studying electron transport chain, bacterial respiratory electron transport chain. See here, whenever there is a energy generating metabolic process, it may be substrate level, it may be oxidative phosphorylation, or there may be photophosphorylation. Actually, these are the 3 modes of ATP generation. In case of oxidative phosphorylation, the phosphorylated compound will be converted into unphosphorylated compound and there is ATP generation. In case of substrate level phosphorylation, phosphorylated compound will be converted into unphosphorylated compound and ATP generation occurs. In oxidative phosphorylation, one compound get oxidized, other compound get reduced and there is a energy generation. In photophosphorylation, in photosynthesis, you can say the energy generation occurs in presence of light. So it is a photophosphorylation. So these are 3 modes of ATP generation. But here in oxidative phosphorylation, what happens actually? One carrier get oxidized, other carrier get reduced. So there is a simultaneously or alternatively oxidation reduction of electron carriers. So here we are studying what are different electron carriers in electron transport chain in bacterial cell. So firstly we will discuss oxidative phosphorylation. It is simply respiration. It is an ATP generating process in which NADH means nicotinamide adenine dinucleotide. It is reduced one, so there is a H. NADH molecules are oxidized. When it is oxidized, H will be removed and NADH will be converted into NAD. So NAD is the oxidized form, NADH is the reduced form, and electrons are transferred via series of carriers to reduce the final electron acceptor. It is inorganic molecule. Final electron acceptor depends upon type of respiration, whether it is aerobic or anaerobic. If respiration is aerobic, final electron acceptor will be oxygen. If respiration will be anaerobic, final electron acceptor will be other than oxygen. It may be in any inorganic molecule. It may be carbonate, nitrate or sulfate, what it may be. So whatever electrons and protons are transported from one carrier to next carrier, ultimately it will be accepted by final electron acceptor. Either it may be oxygen or it may not be oxygen other than oxygen. It may be carbonate, sulfate, nitrate. So that is the respiration or that is oxidative phosphorylation. These carrier involves in electron transport chain are arranged in particular sequence. You should remember that the sequence of carrier depends upon their potential, electronegative potential or electromotive potential. So depending upon that, they are arranged in particular sequence. The first will be NAD, then will be FAD, then coenzyme, then cytochrome, and then terminal electron acceptor. So this is the sequence of electron transport carriers in electron transport chain. It depends upon their potential. So they are arranged in particular sequence in plasma membrane or in mitochondrial membrane. Plasma membrane in bacterial cell, mitochondrial membrane in eukaryotic cell. This arrangement is called as electron transport chain. Now, oxidative phosphorylation occurs at the cell membrane of bacteria and at inner mitochondrial membrane in mitochondria in eukaryotic cell. It is the major process considered in bacteria, and the arrangement of electron transport chain in bacteria is same as that of the mitochondrial membrane. So we will study in next lecture regarding the electron transport carriers NAD, FAD and coenzyme Q or quinone, and how it is oxidized, how the carriers get reduced one, and how there is a energy generating metabolic process, how ATP will be generated. Thank you.